Good afternoon, and thank you for joining us for the Outdoor Art Club presentation of one of the big guns of fighting the coronavirus. I'm Robin Geisler, and I'm working with fellow OAC team member Lynette Dennison to bring you today's program. As you know, we're using the Zoom platform. We found that Zoom is not always perfect, and neither are we. Our team has worked hard to master everything that Zoom has to offer, but things still don't always work out quite the way that we think they will. Please be patient with us. For those of you who are not familiar with the Outdoor Art Club, we're a nonprofit women's organization founded in 1902. The club engages in civic, cultural, and charitable work in our community. To give you an idea of some of our past contributions, several years after its founding, the Outdoor Art Club provided food, shelter, and clothing to the victims of the 1906 San Francisco earthquake. This past year, club members provided 100 holiday gifts for women and children at the Center for Domestic Peace in San Rafael. And every year, the Outdoor Art Club runs a speaker series so that people like you can learn more about topics as varied as organic gardening and the latest research on our battle against the coronavirus. Before I turn this program over to today's program chair, please note that Zoom has two different functions, one for chat and one for Q&A. We won't be using the chat function today. However, we'd like you to use Q&A to send your questions to Dr. Ott anytime during the program. She will leave time to address your questions at the end of her presentation. And at this time, I'd like to introduce Janice Prochaska. Janice is a member of the Outdoor Art Club and program chair for today's event. And I don't know if Janice is on screen, but hopefully she will appear soon. And thank you, Janice, for bringing us today's very timely presentation, one of the big guns fighting the coronavirus.
Welcome, everyone. Our speaker today is Dr. Melanie Ott, who was born and raised in Germany. In the early 90s, she led the IC unit at the University Hospital in Frankfurt, where 90% of the patients were HIV positive. All died due to no known treatments being available at that time. That experience drew Dr. Ott to the study of virology in Germany and later in the United States. In 2002, Dr. Ott joined Gladstone Institute, a biomedical research institution affiliated with UC San Francisco, in that Gladstone Institutes, there are 5 major research institutes. In April of 2020, just about a year ago, 1 month after we were all sheltered in place, Dr. Ott was featured in the San Francisco Chronicle as one of the big guns fighting COVID-19. Those big guns included doctors, scientists, infectious disease specialists, and software developers. 1 month later, in May of 2020, Dr. Ott became the director of the 5th Gladstone Institute that focused on virology. This institute searches out rapid diagnostic, prevention, and treatment strategies for infectious diseases. It is the hub of virology research in the Bay Area. I especially appreciate Dr. Ott's commitment to collaboration to advance science. Our format today is my asking a few questions of Dr. Ott, with her responding. Afterwards, we will open up to your questions. To start, Dr. Ott, could you share with us what you do at the Gladstone Institute?
Thank you so much, Janice, Robin, and the whole OAC team. It's really a pleasure to be here. I'm also a fellow Millvallian, living here since 10 years and really delighted to be here with you today. I will share my screen. And so here, this is our building, the Gladstone Institute building in Mission Bay at the Mission Bay campus of UCSF. And what we do is we are overcoming unsolved diseases through transformative biomedical research. We work on the heart, the brain, and we work since more than 30 years also on the on viruses. So this is my institute, the faculty of my institutes. And if we're talking about big guns, I'm probably a small one compared to somebody like Jennifer Doudna, who is having a lab right next to me on the 5th floor of our beautiful building, and working with us on diagnostics and CRISPR technology. But all my colleagues have pivoted part of their research last year to SARS-CoV-2, and I think we have made amazing progress and contributed meaningfully. The first thing that we had to do is to build the appropriate laboratory to culture the virus and to work with it. So while we have been working with many viruses in the past, especially HIV, hepatitis, Zika, influenza, and herpes, we were not equipped to work with a respiratory virus like a SARS-CoV-2. So we built an additional high biosafety laboratory at Gladstone, which allows us now with the appropriate respirator and protective gear to work safely with this virus. And there's about 10 to 15 researchers in this laboratory around the clock. Now, this laboratory really allowed us to look into this virus in much more detail. All viruses are inherently incomplete. This is really one thing that we have to keep in mind. They are not a complete living, they are a semi-living creature. They need a living host in order to survive, to multiply, and then to spread to the next host. And that's really something that we have been interested in at the Gladstone Institutes for many years, exploiting this dependence of viruses on the host cell. And I'm showing you here different cells, the lung cell, the primary host for the virus, SARS-CoV-2, which is the causative agent for COVID-19, but also gut and the heart cells. And you see each of these blue dots is an individual cell, and each of the green dots down here represents a replication center that the virus had set up in these cells in order to replicate and multiply. And you see that some cells, especially the heart cells, do very poorly with the virus replicating because the virus basically kills it as soon as it deprives it from all its resources. So this focus on this interaction between the host, us, and the virus is really a long-term focus in the Institute. And especially my colleague, Nevin Krogan, has really brought this to perfection because he has been studying since years all of the host proteins or the host factors that get in close proximity to the virus once it comes into our cells. He does this with a specific technology called mass spectrometry, but basically you query all the 20,000+ genes that encode for proteins in our body and see which ones are in direct connection with the SARS-CoV-2 virus or other viruses before. And then he builds these very intricate maps where we can identify already last spring in a record time 332 proteins that directly interact or make contact with viral proteins in a cell. And just by looking at these host factors that were identified, we could identify already 70 drugs that we predicted are effective against in suppressing the replication of the virus in our cells. And these are still under investigation as potential antivirals. Another approach that we took last year was with my colleague Andreas Pushnik at the Jen Zuckerberg Biohub, which is in very close proximity in Mission Bay to us. What we used here is instead of looking which of the 20,000+ genes or proteins are in contact with the virus, we just knocked them all out one by one, all of them, and then asked which of these proteins are required so that SARS-CoV-2 can still replicate and set up shop in our cells. So we did this knockdown using CRISPR technology, of course, pioneered by my colleague Jennifer Doudna, and then identified a set of genes that were absolutely required, that basically that we provided, that provided help from us to the virus in order to replicate. And we not only did this with SARS-CoV-2, we did this also with 2 other coronaviruses, common cold coronaviruses that are seasonally circulating in all of us every year. And we found a series of common host factors that were not only necessary for SARS-CoV-2, but also other coronaviruses. And these are the gold. For us, this is gold that we found, because these common pathways are exactly the ones that we have set out to target. We want to find these common pathways, the common lifelines that we are throwing the virus with in our cells, in order to develop drugs that would not only be effective against one pathogen, here SARS-CoV-2, but would actually be effective against potentially all coronaviruses in this case. And that would also include a coronavirus that will come around the corner in the future. Our classical drug design is different. We have made beautiful drugs, or biotech companies have made wonderful drugs against HIV and hepatitis C. These are highly specific against one pathogen. And often, because the pathogen, as we know now with the variants, can mutate and can start developing resistance, one drug is often not enough. So we have actually to combine multiple drugs against one pathogen, which is time consuming and expensive, versus having one drug that might potentially protect us and help us with multiple pathogens. So this is really one of the foundations of the Institute that we are working with.
That's amazing to have one drug to help in so many ways, and it would be really great for our future. Could you help us understand what we could do better to battle COVID and future pandemics?
Yes. Well, I think the key is to get the vaccine now that it is available. The second thing is that we still have to practice our social distancing and masking because of various reasons that we can get into detail. But the third thing is really testing. I am very passionate about testing, and we have seen in this pandemic how absolutely critical it is, because contrary to the SARS-1 that had an outbreak a few years back, SARS-CoV-2 is different. It spreads asymptomatically. So we cannot identify people just by taking temperatures and then isolate them and break the pandemic. I think what we have to do with a pathogen like SARS-CoV-2 is to test frequently, to have a fast test result, and to have it everywhere available in order to proactively, constantly test in order to get ahead of this virus. Now, my colleague Michael Mina at Harvard has modeled that if we would test every day or even twice a week, and we would have our results immediately or in a very short turnaround time, and can then isolate depending on a positive signal or a negative, we would have 0 infection rate in this country. So we have been thinking about this type of test, not just since last year. My colleague Jennifer Doudna and Dan Fletcher, both from Berkeley, but also both affiliated with the Gladstone Institute of Virology, we have been thinking about this already since 2 years. And I think we were initially thinking to develop a diagnostic that would be useful at home for HIV, because more and more people are now getting off therapies. They're trying different therapies. And that requires that somebody is taking their viral loads. They're measuring the amount of viral RNA constantly in their blood in order to go back on medicine if necessary. So we were starting out with HIV, but pivoted the technology in last January to SARS-CoV-2 because it's also an RNA virus, and we could easily adapt the technology, and I show you why. Now, in order to be at home or to be available everywhere, we turn to the mobile phone because we know that it has a lot of features, including the camera, that can allow molecular testing. And I show you in a minute how. It is a very sensitive measurement. It is portable. It's a portable device. It is trackable, and it's existing and widely available. So we were building a test that would directly use the camera of the mobile phone to read the outread. And the phone was the computer, the GPS to do biosurveillance, or to upload it to the cloud for storage and analysis. We combined the mobile phone with the new CRISPR technology that Jennifer has brought to us. And specifically, we looked into an enzyme that's called Cas13a, and that is specifically recognizing RNA. And I told you that RNA is the genetic blueprint for the SARS-CoV-2 virus. Now, if we use this enzyme and we just give it a little guide, a little snippet of the virus, this enzyme can find the real SARS-CoV-2 genome in a sample and then gets turned on. And when it's turned on, it starts cleaving, and it cleaves a reporter that we supply into the reaction separately, and that starts then eventually, when cleaved, it starts to emit fluorescence or it glows. So it's an extremely simple and rapid test, which does not require, like PCR, that we turn the RNA into DNA and then do amplification of the signal and then start detecting this amplified signal. We optimized the technology so that it would work just with these 3 ingredients, including the sample. And what it does is we built this homegrown device, which we called Rovir. We were inspired by the Mars rover, and we thought that it would break new frontiers. And you can see that the phone is basically lying on this box where we enter the reaction on the side, and the camera is really measuring this fluorescence that is emitted once the sample is positive. And we can see this within minutes, and it becomes more and more intense, but the algorithm that we wrote in the program can detect it after 5 minutes of analysis, and it can distinguish between a very positive and a less positive signal. So what we extremely excited about is that this technology not only tells us whether there's RNA there of the virus, yes or no, but it also tells us how much is there. And we think that this has profound clinical implications. Now, the 2 secrets that we found were important for making this work was that we could not just rely on one of these little snippet guides. When we did one, we were not in a sensitive range that would allow us to use this as a diagnostic. When we started to use multiples of them and lined them up along the genome of the virus, we found that we were very rapid and very sensitive in our detection. And the second surprise came with the mobile phone, because usually we run these assays on a $25,000 instrument, a plate reader in the laboratory. And we thought the phone has to be as good as the plate reader. Let's hope that it is as good as the plate reader. Now it turned out that the phone is actually 10 times better than the $25,000 plate reader, because as you can just simply see here in this curve, it is much less noisy. And so the way that we have, that we are enjoying better and better quality with the pictures that we take with our mobile phones, it has also more and more improved the sensitivity and the reading capacity of these cameras. And so what it allows us is to actually call a sample much faster positive than when we have noisy reads like with a plate reader like this. So we are extremely excited about this technology, but I showed you this is all homegrown. It was built by a student in Dan's lab, handmade, hand-aligned, and it was really amazing to watch how fast the technology came together. Now we're working with outside partners to turn this into potentially a device that can be used in the community. What it has to fulfill is we cannot rely on any laboratory steps in between. for the device to be really at your home with your mobile phone or at a point of care at a doctor's office, it has to go from end to end, from the nasal swab, put it into the cartridge, and then everything else is done by itself, read by the mobile phone, uploaded and analyzed into the web. And that's where we currently are. And it was a little bit harder because it was not just us who needed to be coming on board. We have to now rely on our partners, but we are very close to having a end-to-end device that we can use, where we can use the mobile phone to read this without any laboratory involvement. And the interesting thing is, and why I think this is important for the future, is these diagnostics are new because and they're extremely versatile. As fast as we were moving the test from HIV to SARS-CoV-2 in January, as fast we can move it now to the variants, and we can move it to other viruses like influenza or others that we aim to detect in the same swab to see if somebody has the flu or somebody has COVID.
That's really amazing, all the work that has been done in just a year. Can you share with us how soon you think this would be something that people could use in their own home?
Well, yeah, we had hoped we would have it already in our own home. I think we got a little dose of reality and how long things take to actually translate once you are not the only one responsible for it. But I think we are close to having, I would say, in the next 1 to 2 months, a device that we're going to start to test for FDA approval. And then that opens the door for manufacturing and distribution.
And you know how you were talking about having many tests being done all the time. Would this be something that we would have to take into the future and test ourselves for other kinds of things?
Well, I think it is sort of a principle. It shows the principle. What we learn with SARS-CoV-2 now and all the investments, they will be also useful for other viruses of the past, the present, and the future. And I think that is what is exciting. What I find exciting is that I just use my mobile phone that I have with me all the time, and eventually might be able to use it as my diagnostic. So I brush my teeth in the morning, I take my test, and then I go out in the world or I stay home, depending on my test result. I think that's my vision, to make a consumer electronic adaptable as a diagnostic. And I suspect that the need will be there because we have all become now so aware of viruses and how dangerous they can be. And I think I think all of us want to have more access to more frequent testing in order to make sure that that ourselves, but also those that surround us, are safe.
Thank you. Could you tell us some about the different types of vaccines that are available now and how they differ?
Yes, the vaccines. That's the big question. I think I have to really point out that we have currently more than 60 vaccines coming down the pipeline in various clinical stages. We have 2 approved in the United States, and this is, in my mind, an enormous triumph of science. And we have different types of vaccines. So we don't have just one type fits all. I think we have different types that are either coming close to approval or coming down the pipeline. I want to just point out that I'm sure you all know the Pfizer and BioNTech and the Moderna vaccines. These are RNA vaccines, relatively new way of making vaccines. And what that means is that we're not generating the piece of the virus in the laboratory and then give it to people. We basically give the genetic blueprint, the RNA, directly into the into people, and then we ourselves basically produce the protein that will then train our immune system to recognize the virus. And that, of course, is not the whole virus. This is just a little snippet again. And what we all focusing on is the glycoprotein, the protein that is on the surface of the virus and that makes the contact with our host cell in order to get entry and then hijack the cell. That's the so-called spike protein. So in the RNA vaccines, they're basically using the blueprint, and we're injecting this as a little nanoparticle. The problem is that those are very finicky in the way that they need to be kept very cold. And that's not so much the RNA that needs to be kept cold, it's actually the nano particle around it. And I suspect that we will see a lot of innovation in this in the future to avoid this dependence on the cold chain. The next type of vaccines are the AstraZeneca and the Johnson & Johnson, and also the Russian Sputnik vaccine. These are so-called vectored vaccines. So instead of giving the blueprint, the genetic blueprint directly into the arm, we piggybacking the blueprint or the genetic information of the SARS-CoV-2 spike protein onto another virus, a harmless virus that has been inactivated, in this case an adenovirus, also a respiratory virus. And that vector basically shuttles that into the cell and starts the production of the spike protein, which then alerts our immune system so that if the real virus comes around, we are prepared and can get into full action immediately. Now so the AstraZeneca has not been approved here in the US. It has been approved in the UK and also other parts of the world. It has had a little glitch in its rollout because during the trial, accidentally, there was given half of the dose in one arm of the trial instead of the full dose in one of the shots. It turned out this was a good thing because that increased the efficiency from 70 to 90% protection, but the FDA is not accepting this and want them to go back and perform a regular trial. Now, Johnson & Johnson just revealed their data, very good data, also 72% effective. The advantage here is that they're only giving one shot. So if they would do two shots like everybody else, we suspect that the effectiveness and the protection would go up into the 90+ range also. But having a one-shot vaccine that doesn't need a super cold chain and is 72% effective and had also some effectiveness against the variants, I think is a very good news for us and the rest of the world. So these are the vectored vaccines. The Novavax that you might have heard about is a different type of vaccine. It's called a protein subunit vaccine. That's more old-fashioned. That's what we do. We isolate the spike protein in the laboratory and then inject it and then alert the immune system. That had very good efficiency also with 90% protection, but also a little bit of a diminished protection against some of the variants. And what is not shown here is the fourth version, which is actually the oldest version of a vaccine where you use the whole virus, you inactivate it so that it cannot be infectious, and you inject it into people so that the immune system gets alerted. This is the Chinese way to go. They have generated multiple whole virus vaccines, which are currently not approved here in the US. So you see 70 to 95. I want to remind you that we started out with the expectation to have a vaccine that might be more than 50% effective. So all of the vaccines so far have definitively exceeded our expectations, especially the RNA vaccines, because they are really in an extremely high range. Also, especially in the elderly, where we know that the immune response is usually a little weaker. But what I wanted to point out with this slide here is that all of the vaccines, regardless of what they were made of, have provided 100% protection from hospitalization and death due to COVID-19. And I think this is really the message that we should keep in mind whenever we think about the vaccines, that they are 100% effective in keeping us out of the hospital.
Doctor, what are your concerns about mutations in new strains that are coming up?
Whoops, I'm very concerned about new strains. They're keeping me up since December when we have first heard about the UK strain, and you might have heard about that too. I just wanted to point out variants of viruses is something that is very natural for viruses. Viruses constantly make mistakes. They accumulate these mutations, and they do this by purpose, in parentheses. because that helps them to adapt, because they can then see if there's anything coming their way, like an immune response or a drug. They can then select a variant, or a variant will be selected that by chance has actually a benefit and will outgrow and take over. So this is something that is very normal and was expected for this virus. The coronaviruses in general do not build in very, very many variants. They are actually pretty confident in the way that they reproduce their genomes with very little error. However, they do make error, and when they make an error, it can lead to a variant. So what we are dealing with, we have thousands of variants of this virus already in the books. However, now we're dealing with variants of concerns, so-called VOCs. And I think they have started to arise last year, end of last year. And there's two things that I want you to take away from this. They have arisen in areas where we have very high replication. Variants, when the virus has a chance to replicate, and it replicates in many, many more people than in other places, variants have better chances to arise. So it's no surprise that the UK, Brazil, South Africa, and unfortunately California are areas where these variants of concerns are arising. The problem why they are variants of concerns are because they are more transmissible, and this was really first shown here in the UK variant, where end of last year the numbers of the new cases or confirmed cases were going down in the UK, but all in a sudden they experienced this enormous spike. And this spike in new cases was really caused by this new variant. We have calculations now that the UK variant is going to take over in the US in the next 1 or 2 months as the predominant variant, because it is the one that is easier transmissible. And then giving you a little bit of virology 101, and I want you to remember these numbers. The 501 is the position of the mutation in the spike protein that characterizes the UK variant. It is also found in the Brazilian and the South Africa variant. And that is a position in the spike protein that makes the spike protein latch onto the receptor and onto the host cell much more effectively. And that's why this virus is easier transmissible. It also is causing likely more severe disease simply because it replicates to much higher levels. The good news about this variant is that it's almost 100% covered by the current vaccines. So we don't have to worry as much about this variant, but we worry a lot more about the Brazilian and the South African variant. And the reason is that they not only have the 501 mutation, they also have an additional one, the 484 mutation, which is adjacent to the 501. But this is the mutation that helps the virus to evade the antibody response, either by people who had been infected before or who are vaccinated. And so this is diminished in the 484, not completely, but in the Johnson & Johnson, we know that it's about 57% effective in South Africa, where the variant is dominant. So it lowers the effectiveness of the vaccine. And that's why the reason why Moderna and also Pfizer now modifying their vaccine to potentially include a booster where we specifically immunize against that 484 mutant. Now, California is special. We have our own variant here, not connected to UK, Brazil, or South Africa in the regular or unifying mutation number. So we have a different number. We have 452, which is here. And I show you in a minute why this is concerning. This is a mutation that's also in the spike protein and the so-called receptor binding domain, where it really latches onto the receptor. And this is a variant that is close to the MINC mutation that you might remember in Denmark a few months ago, when all colonies of minks were slaughtered because of this mink mutation, because it is highly transmissible and it was coming back from the minks to humans. So this is also a highly transmissible variant. Also, it does not have the 501 mutation that I showed you before, but it is the variant that's responsible for the very high attack outbreak at Christmas at the Kaiser Santa Clara, where almost 80 people were infected by one person. We don't really fully know how well it responds to the vaccines. This is under very intense investigation. We're growing that variant currently in the laboratory that I've showed you in the second slide. And we're working very, very intensely with a consortium of all virologists and BSL-3 facility owners in the area. We're working with UC Davis, we're working with Berkeley, we're working with Stanford, we're working with the Blood Center Vitalant here, and we're working with UCSF and Gladstone, really at all angles and record speed again to find out how dangerous that variant is. The thing that we know from my colleague Charles Chu at UCSF is that it is persistently increasing in frequency. So we have now, we know now that about 50 or more than 50% of the infections in Southern California are caused by this variant. And in here in the Bay Area, it is in around 20 to 30%. So I think this is really something that is an evolving situation. And the key here for us is that we're winning the race between vaccination and a variant. We have to make sure that we have a buy-in from the population and a fast rollout of the vaccine in order to bring our numbers down. The key is that our numbers, unfortunately, are still out of control here in the US. And I think that is the breeding ground for variants. And we have to make sure that we continue to be vigilant, we get the vaccine, and we continue to practice social distancing in order to get these numbers down so that the variants cannot arise.
Can you share with us, though, if we get the second shot and we wait 2 weeks, do we have any more sense of freedom than we do at the moment? Can we invite vaccinated people over for dinner? Can we take an air flight?
Yes, these are all very good questions and very pressing questions. I think currently the recommendations are that we, even if we are vaccinated, we continue to practice social distancing and masking and testing in order to avoid infection of people who might not be vaccinated or might not have a very strong vaccination response. The problem is that the 94% effectiveness that I quoted of the vaccines is for symptomatic disease. It's not for severe disease. It's not for asymptomatic disease. So we don't really know precisely whether the vaccine prevents us from becoming infected, even with the wild type virus. And the estimation based out of Israel, where they have a very high vaccination rate currently and are testing like crazy to make sure to know whether there's asymptomatic transmission still, is that the RNA vaccines are about 60% effective in suppressing asymptomatic infection. So this is a very good number, but still it leaves 40% open that we might be infected and carrying and transmitting. Now, if other people are vaccinated, the chances are lower that they become infected, but we have still a level of uncertainty currently in how the variant, especially here in California, is going to respond to the vaccine. So there is still a possibility that we can become infected with a variant despite being vaccinated, not having much symptoms, but still also transmitting it to somebody who might develop more symptoms. So I would be cautious for now. I think the time is coming soon where we will be able to loosen our guard. But I would say now is not the time, because we are in this race between the variants and the vaccine. And I would say the more we can wait a little bit and still become and still practice social distancing and with outdoor dining and other things, I think it would be very beneficial, because we are in a good position to win, but we don't want to weaken our position currently.
My last question to you before we take those from the audience are, what lessons should we learn from the current pandemic for the future?
Yes, there's a lot of important things that we should keep going after this pandemic. After hopefully we have overcome this pandemic, I would say a very important thing is we have to make sure that we have a coordinated federal response in place here for the U.S. because I think that clearly slowed us down. I think we have to make sure that we are involved in the worldwide community. We should not leave the WHO during a pandemic because as my neighbor Larry Brilliant and fellow Millvalian says, if it's anywhere, it's everywhere. So if we are not vaccinating the whole world or making sure that everybody is, because that immunity is going to be arising everywhere in the world, we will get the variants that arise somewhere else back here. So I think that's a very important lesson that we should keep in mind. And I would say third is we have to invest and trust into science. I think these vaccines and I think the advantage that we have seen last year have really underscored that we in the scientific community, we can work together and we can make things happen fast. And for me, the spirit of collaboration that I've experienced during this pandemic is really mind-boggling. I think everybody was a virologist, and my colleagues at Gladstone who are cell biologists, stem cell biologists, they all offered help. They all offered their expertise. Worldwide, we exchanged reagents, information, and especially the Chinese scientists at the beginning who posted the sequence of the virus, they really jump-started everything, and that was a heroic act.
So some questions from our audience. We wondered, how did you end up living in Mill Valley?
Because it's the most beautiful place to live in the Bay Area. I think we lived for 10 years in the city. Both my husband and myself, we were working at UCSF and at Gladstone. But we ended up every weekend in Mill Valley or around Mill Valley and spending our times on the beach or hiking or doing other things. So eventually we made the jump while still both working in the city, and people thought that this was really odd. Why would you move out and have such a long commute? I have always said that the commute, I have the luxury to commute in the most beautiful place in the world, the Golden Gate Bridge and Marin Headlands. I think it cannot become much nicer. And eventually my husband, Eric Verdin, became the president of the Buck Institute, and it made total sense for us to live in Mill Valley because he's basically commuting to Novato and I'm commuting to Mission Bay. But I feel blessed to live in Mill Valley. Our kids love it, and it could not have been a better decision.
There's a couple questions about the testing device, like what would be the price point for the testing device, and could somebody sign up to volunteer to be in part of the testing part of it, the testing phase?
Thank you very much. Yes, so we're trying to keep the price down as much as possible. We have worked with partners in India who have very inexpensive phones, $30 phones that also work with this device, so it doesn't have to be a very high-end phone. Google has been extremely generous with us, offering us especially scientific support in the way that they opened all their books up for the optics of their Pixel 4 phone so that we could completely align our instrument and really learn what was necessary and what was not necessary. They have been very generous also providing us with phones, and they offered that they will distribute phones for free in order to combine it with the instrument so that we don't have to build at the beginning a version for every phone, because that's the problem with the mobile phones, that we currently building it basically for the Pixel 4 and then later adapt it. So yes, the cost, I think we hope to have a cartridge that is under $10 in terms of costing, or even cheaper. And I think the device, depending on whether the phone is being used or the phone has to be supplied from elsewhere, I think is probably less. It's in the $100 range. But we're trying really to get this further down. It all depends on how the final design will work out. In terms of testing, we would love to have as many volunteers as possible. We're currently working with UCSF and also with Berkeley, with the IGI, where Jennifer Doudna has set up a testing lab to test our device in parallel with the regular PCR technology that is currently been explored. So I think this will come soon, but we're not yet quite there.
Okay, another person is wondering what could happen if the second shot is delayed beyond the 42 days time frame.
So I think it's an interesting question, and you might have followed up that Astra had recently a trial where they delayed their second shot for 12 weeks, and it still worked. It actually worked a little better than before. So I think that the timing between the first and the second shot, I think, is not yet set in stone, and we will find out. I think what is important to keep in mind is that when you give the first shot, your immune system starts to ramp up and builds basically the memory of the viral infection that will help it when the real virus comes in to react very fast. The second booster is basically meant to cement this memory so that it becomes more longer lived and more robust for the second shot, for the reinfection. So I think the timing is important in that way, that I think we don't want to lose the first wave of immunity, because then we have to basically start from zero. I think you want to have the second shot in an area where you can really still have the first wave of immunity strong enough so that you just need a booster to get it up into the real protective realm. But I would say the last word has not been spoken about what is the timeline between first and second shot. The question is, will we have a third booster because of the variants that will be required? But I would say these are all good problems to have, because we have the luxury of having already 3 types of really robust and very interesting vaccines that I think we can build on and make even better.
Could you share a little bit more about how you got into the field that you're in, really the focus on virology?
Yes, I'm glad to do this. I'm having a little bit of a zigzag career, because I really started out as a very devoted clinician in Germany in the 90s. And you might recall that Frankfurt was one of the very early epicenters of the AIDS pandemic at that time. It was San Francisco, Amsterdam, and Frankfurt. We even had a Frankfurt nomenclature about how we created the different symptoms. And it was a very heartbreaking time in the way that initially we didn't know what the disease was. Then we had nothing at hand to help patients. And when they came to me, because I was responsible for an intensive care unit of about 6 to 10 beds, I think they usually had problems in their nervous system and were terminal. So it really inspired me. And it was not only my patients, it was also my nurses who became infected and others. So it was really a heartbreaking time. So I decided I wanted to take a 2-year out time in the way that I wanted to really dig deep into the biology of that virus, learn more about it, and maybe find ways to help in some way. Now, in the mid-90s, we got the protease inhibitors, which changed the disease from a deadly disease to a manageable disease. But still, it took a while to get to the status that we have today, where people take or even get a shot every 3 months and are fine. So I think an amazing victory of science here with HIV. But at the beginning and the time when I worked on it, really a similar urgency that we have today to find something that could help. Now, why I did not go back to the clinic as I previously planned, I wanted to do just a 2-year postdoc in a laboratory, get familiar with the virus, and start, getting familiar with molecular technologies. It was the time of molecular medicine, really exciting. But what got me hooked with viruses was really the finding that these viruses are so intimately intertwined with our own biology. you just have to follow a virus into a cell and you find very interesting new biology, because they go really after our nodes of most importance. And so that is what got me hooked. And that's why I have never returned to clinical medicine, and have then started a career in biomedical sciences, just because I always felt that the next discovery was just around the corner and I needed to continue. So I went from New York to Heidelberg and worked there in the cancer center together with Harald Zurhausen, who got the Nobel Prize for finding that papilloma viruses cause cervical cancer, and also being instrumental in the vaccine that we now give to girls and also boys. And I think these are all inspiring stories that really show you how a discovery can lead to something that that can really benefit people and humanity. And so I am personally extremely excited by viruses. Also, they are of course bad for us, but I think they tell us also enormously about ourselves. And many of the groundbreaking discoveries that have been done in cancer and in other areas have been done with the help of viruses.
So someone is saying here that COVID vaccine stimulates the immune system. Should a delay in getting the vaccine be a consideration before or after a person has major surgery, for example, hip replacement, which likely also stimulates the immune system?
Yes, that's a very good point. I think it's correct. I think if you have a surgery or have just undergone a surgery, I think you should consult with your doctor. The key point is less the immune stimulation that you have during the surgery. I would not be concerned about it. I think the key is that you actually are not immunosuppressed during your surgery, and your immune system cannot mount the right response to the vaccine. So I don't think, I think I would highly recommend that you consult with your physician and determine the best time when the vaccine should be taken.
What do you think of the CDC recommendation that was in the press today, that after 2 doses of the vaccine, it is unnecessary to quarantine if you're exposed to COVID?
Oh, that's interesting. I haven't seen this, but I think it's a sensible step because there is protection by the virus, and I think at the same time CDC is also recommending that we continue with our mitigation strategies. And I think if we do this, I think the risk is relatively low. I think CDC has been very proactive in shortening quarantine in the past. I think there was a huge problem for clinicians, especially, that if they got into contact with somebody who was positive, people had to stay home 2 weeks. That, of course, was extremely disruptive for many of the clinical services and others. So they have been very aggressively shortening the quarantine time in the past. And I suspect that this is sort of a continuation of this policy where they're really trying to make sure that that our health professionals don't have any unnecessary downtime.
Why do some people have a reaction to the vaccine and others do not?
That's a very good question. I think that's one thing that we will learn further in the future. I think there is one serious side effect that has been reported is a so-called anaphylactic shock, which is a severe allergic reaction, which has occurred so far as I know, only in 2 people worldwide. We have now vaccinated about 138 million people. And this is why we all staying 15 minutes after the shot there to be monitored. So this is something that is completely treatable. And I think this 15 minutes is the safety measure to make sure that nobody goes home and then has a severe reaction. And as far as, I mean, I just got my 2nd shot yesterday at San Francisco General Hospital, and I was chatting with everybody there, and they told me that they have never seen it in the whole vaccination time that they have been giving.
Is Gladstone doing basic science or pursuing patents?
Gladstone is doing basic science. As I said, we're doing basic science in the realm of heart, brain, virology, and immunology. We also have an institute that is led by my colleague Katie Pollard, and that is specifically dedicated to big data science and technologies. I think in general we are doing discoveries, and if it's necessary, and if it's furthering the discovery, we also do patent, and because it is a necessary step to potentially market and manufacture a device like the diagnostic. So we do patent, but we only patent in order to further the technology and to allow it to potentially become widely distributed.
This is a series of questions about boosters. Do you think the Moderna booster would be ready this year? Would vaccinated people need to get the booster within 12 months of receiving the vaccine? Can vaccine boosters be interchangeable? For example, if you had the Moderna 2 shots, could you get the Pfizer booster?
Yes, very, very, very good questions where we don't have a firm answer yet. I don't know how fast Moderna can pivot, but I would suspect that they can pivot very fast and can have a new booster ready in a matter of weeks. So I don't think that this is a problem. I think it sort of speaks to the broader question, which is, are we going to have boosters regularly in the future? And you heard from the Moderna CEO who said that he can envision that certain parts of the population, maybe particularly the elderly, might have to be boosted more often than younger people or other parts of the population. But it could be that we will have to adapt to certain variants similar to the flu vaccines and make sure that we are vaccinating against those more frequently. Now, in order to mix and match, I think we will have to look more into clinical trials to see and find out whether that is safe. There is no obvious reason not to do it. But I think in general, the FDA doesn't like sort of mixing and matching. And I think if we want to do this, I think, and as a scientist, I think I would prefer to see some data before we start this.
Does the 484 variant show up anywhere else besides California?
So the 484 variant is actually very unusual here. We have low numbers of the 484. Remember, that was the Brazilian and the South African. Here, our variant is the 452, which is the one that is unique to California. And I have not seen this being picked up in other places than in California. And it is, currently spreading. However, I have to say that many of these variants, we can actually induce the virus to mutate in a certain area, in a certain direction, when we treat it with antibodies in the dish. So we can predict which mutants are coming up. And the 484 is one that always comes up in all of these selection experiments. The 452 is a little bit more rare, but it has been picked up in these like forced studies. So we are careful. We think that it could arise also in other areas or spread to other areas.
And I think this is the last question since we're getting close to 2 o'clock. Long haul COVID. This person has read that long haul COVID occurs more after mild disease than severe. If vaccines still allow mild disease to occur, is there a chance that there will be a higher risk occurrence of long haul COVID?
Yes, very good question. The answer is I don't know, and I think nobody knows currently whether this is a possibility. However, I would say that my expectation is that the vaccines, especially the RNA vaccines, will be efficient enough to actually also prevent asymptomatic disease, so that this is not a factor. I think the long haulers are we have to look very, very detailed into the genetic background of individuals here, and seeing how this genetic background matched with, the viral disease, and why this is causing these protracted diseases. My colleague Steve Deeks in UCSF has started a cohort of long haulers, and I think we will learn a lot from this, and also now from the first vaccinated individuals, whether there is a risk here. But I would still emphasize that I think the 60% effectiveness of the RNA vaccines in preventing asymptomatic disease is a very good number.
Well, Dr. Ott, I want to really thank you for sharing with us today your knowledge, your science, and your research. You are a great example of the power that a woman can be in leadership and service.
Well, thank you so much, Janice, and everybody at OAC for inviting me. As I said, I'm delighted to be here in Mill Valley, and I hope that this was a little helpful for everybody to navigate the vaccination jungle a little better. And my last advice is go get your vaccine. Thank you.
Yes, thank you. And people have asked if this talk is being recorded, and it is with Dr. Ott's permission, and you can view it in about a week on the Outdoor Art Club public events section. I just want to share with you too that a couple more webinars that we have lined up. Our next public event will be held a week from today, February 18th at 1 o'clock. The title is Creating the Relationships You Want with the People Who Matter, and is being led by Gail Weiner, a licensed family therapist. A week later, on February 25th, we will host a webinar featuring Julie Charles from SF MoMA. The title is What's the Big Idea? A look at understanding and appreciating art. You can register for these and all future webinars at the Outdoor Art Club website under public events. And that's also where you can see the recordings. If you are interested in continuing to know about future public events, please visit the public events page on the website and sign up to be on our email list. Thank you again to everyone for joining us today. We hope that we see you again very soon.